From Microbe TV, this is TWiP, This Week in Parasitism, episode 191, recorded on January 8th, 2021. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dickson des Pommiers.
Hello there, Vincent, and hello there, Daniel.
And from New York State, Daniel Griffin.
Hello, everyone.
Dickson, you insist on saying hello to someone who's not been introduced yet.
He's never going to learn, Vincent.
But I can see him.
Daniel, that's why you're a physician. You're patient. Yeah, he's never going to learn.
Ooh.
That's fine. It's fine.
It's okay. We'll take him as he is.
All right, our first TWiP of 2021. Dixon, Happy New Year.
Yep. Same to you, Vincent.
Happy New Year,
and
Daniel.
to Daniel.
Oh, happy New Year, Vincent. Happy New Year, Dickson. Happy New Year to everyone out there.
Our clinical case for today was presented by Dickson. Isn't that right, Daniel?
This is correct. So this will be interesting.
So it's a 50-year-old male from North Carolina, rural part of North Carolina, suddenly notices blurred vision in his right eye. Went away, hour later, normal vision. Returned. It kept fluctuating between being there and not being there. Finally went to a local doctor, and the local doctor was stymied. He says, but I bet somebody up at Presbyterian Hospital in New York City knows the answer. So he came up to the Eye Institute. And he was seen by Dr. DeVoe, who was the head of the Eye Institute. Mm-hmm. Dr. DeVoe was a good friend of Dr. Harold Brown, who was the parasitologist that I worked for as a technician, then as a graduate student, and later on I had the pleasure of introducing him to our medical students after he retired and I took over the course. So Dr. Brown and Dr. DeVoe and myself went over to the Eye Institute, and we sat down with this gentleman, and they put him on an ophthalmoscope, and they looked into the anterior chamber of the eye in which the vision was good and saw nothing. And then they switched eyes, and they put the ophthalmoscope on the eye that had blurry vision, occasional blurry vision, and they saw something. That's the end of that story. Because when I looked, I knew what I was looking at, and so did Dr. Harold Brown.
All right, we have...
So now we go to the guests.
We have a number of guesses. The first one is from Tim, who writes, thanks for a great show. I think I was called out as a winner. My address is below. Unfortunately, my diagnosis was wrong. If that disqualifies me, well, this week I am close to sure. No, you still get the book. Don't worry. A 50-year-old male kennel and hunting dog purveyor from rural North Carolina had intravitreal dirophilariasis, a likely human spillover of the common canine heartworm spread by a mosquito vector. There are several species of dirofilaria, but Parasitic Disease's 7th edition details Dirofilaria imidis on page 291. Imidis is more likely given the fact the patient raised hunting dogs. Dirofilaria tenuis, heartworm of the raccoon, is also possible. The dogs have to hunt something. This link has a video of the removal of a conjunctival infection, which I have now watched several times, and I'm pretty sure I can't unwatch it. Thanks. As things go, I may never leave my SARS-2 bunker. Here are some images taken from a journal article, which is educational and worth checking out if this guess is right. This is likely what Dickson saw, and he gives some images, macro and microscopic images. And then he wrote, this is curious from that write-up. In humans, the nematode causes a pruriginous subcutaneous nodule, which contains an immature adult nematode. Due to immunological and microenvironmental factors, the parasite cannot reach maturity and is unable to express larvae in the circulation. That's why serology is of little value and eosinophilia is seldom present. If this is the case, could you speculate as to why this is? That must be why Dickson and his associate were not concerned so much with the parasite. Joseph Leidy, who named this parasite, is worthy of his own biography, but I don't see one. Here's his picture, at least. Evidently, he was well-liked and quite a person. As a 23-year-old, Leidy became the first person ever to use a microscope to solve a murder mystery. I would have liked to known him. I'm privileged to know you. If indeed I want a book, I'm going to enjoy every minute of it down to the very last footnote. Please let me know the postage so I can bonus that back to you in the form of another well-earned donation. Take care, Tim. Thank you, Tim. You don't have to pay for the postage. It's okay.
No.
Daniel, can you take the next one?
Certainly. Vanessa writes, Dear TWiP team, as a first-time guesser, I am not at all confident on winning the book. However, I wanted to email a guess at least once before the show gets too popular to possibly read out all the guesses. Like many of your newly acquired pandemic podcast partners, I was introduced to your show via the Michael Minute episode, and since then, I have become an addicted listener to all things TWiV. I have recently completed Vincent's virology course on YouTube and have now moved into Brianne's immunology course. Unfortunately, this order of study means I am still a complete novice when it comes to anything parasitic, a situation I intend to change as a next order of priority. Anyway, here goes my guess. The eyeworm, Thalassia californiensis. Of the 30 known species of Thalassia, only 2 species, Thalassia calipeda and Thalassia californiensis, are known to cause human infection, with the first being geographically centered on Southeast Asia, whilst the latter has been reported to occasionally infect humans in the United States. The clues I used to reach this conclusion were firstly, the emphasis from Dickson on how rare it was to see the entity, and then secondly, the fact that the infected gentleman lived and worked so closely with dogs. The definitive host for these parasites, in fact, these infections in humans are extremely rare, with only a handful described in the literature, and it is generally agreed that human infection is an accidental event. Diagnosis is usually done via the visualization by the clinician, and treatment is by the removal of the worms with fine forceps. However, in this case, it was the early worm that avoided the bird removal. Thank you, Vincent, for all the hard work you put into creating these podcasts. I am a huge fan of Daniel's clinical updates, and I found it immensely reassuring to see the progress being made in treatment of COVID-19 as the pandemic progresses. Dickson, I have to say, you're my absolute favorite. Your cheerful and positive attitude and clear sense of humor is particularly appreciated in these uncertain times. Stay home, stay safe, and keep on educating. Vanessa from the UK.
Well, that does it. She's not getting a book.
I'm going to send her 2 books.
She's from the UK, or that she likes Dickson the best?
No, that she likes Dickson the best. Forget it.
Hey, by the way, Vincent, we just did a show called TWiV in which the server was performing brilliantly. So I don't think it's my server that's causing this delay, but nonetheless, we'll go on. I'm going to read Chelsea. Is that correct?
Yes.
Yes.
Chelsea writes, not enough lotion in the world to fix these hands now. I'm supposed to be doing other work, but could not resist. Of course, after coming up with the answer, I immediately went to rewash my already raw hands, and I'm mildly disgusted by my dog. This will pass in an hour. You describe a man who runs a dog kennel with intermittent, non-painful blurriness of one eye. My guess is ocular toxicariasis from Toxocara canis. The dog or cat have the adult roundworms, and the feces have the eggs. Humans are an accidental host by inadvertent ingestion of eggs, and the larvae can migrate to a variety of tissues. The larvae are unable to undergo any further development, and so their life cycle is terminated wherever they end up, which can cause inflammation of the mechanical damage. That explains why no one else has gotten it. I assume they kept better hygiene or did not work with dogs, infected soil, or feces as much. Although what I am reading describes a lot of inflammatory reactions, it does not sound like he suffered from anything but annoyance. Maybe that's because the eye has immune privilege. I hope it left his optic nerve and perished elsewhere. Otherwise, I would like to think he did suffer some visual loss. Deworming your dogs and cats is supposed to help kill these pests. Picking up their feces on walks also helps, since apparently the eggs can live for years. And of course, don't let your kids eat dirt. Good luck on that one. Thanks for all the work you do, and keep the podcast going, Chelsea.
Leonard writes, based upon the association with dogs and dog kennels and the described conditions of the gentleman from rural North Carolina, I am taking a guess that he suffered from toxocariasis. While it is difficult to confirm the infection since it requires demonstration of larvae via biopsy, and we never got that opportunity with the organism moving over the nighttime hours, and since clinicians were unable to conduct any serologic testing like ELISA or immunoblot to infer diagnosis because it was likely not known testing during the late 1960s with regards to parasites, I still cast a vote for this parasite. The organism likely came by the gentleman handling soil or sand contaminated with infected animal feces. At first blush, I felt this may have been toxoplasmosis, but recalled this is associated more commonly in cats than dogs, yet symptoms were the same or very similar to those of toxoplasmosis. With this being stated, I cast my vote for toxocariasis. I'm a weekly listener to Microbe TV and the various podcasts that focus attention to virology, parasitology, evolution, and the other interesting scientific conversation held with amazing hosts and guests with every broadcast. Thank you to everyone involved in the ongoing truth and education of the public in such important scientific topics. Thank you, Leonard. Daniel.
Kevin writes, I think this is a different Kevin, by the way. Kevin writes.
Right, it's a short letter.
Another Kevin writes, case guess for TWiP 190. Ocular larva migrans caused by Toxocaricatus, most likely acquired by fecal or oral consumption of eggs from some of his puppies. I had a parasite-ridden dream recently in which I led my family through flooded wetlands and, with my dream omniscience, sensed schistosomes homing in on our ankles like heat-seeking missiles. I instructed my family to hurry up and, in the fashion of Dr. Griffin after a dip in Lake Malawi, towel off vigorously once we cleared the water. After our trek through the swamp, I was admitted to the hospital for a cancer, which for some curious reason, they treated with IV-infused mefloquine, an anti-malarial. I know this isn't this week in psychotherapy, but I hope you enjoyed hearing what my brain concocts after reading too much about parasites, dreaming of winning the book, Kevin.
Yeah, it's great. It's very nice. Dickson.
Akara writes, good evening from a suitably dark and depressing Stockholm. Hope you are all doing great and are all keeping safe. As we and I are still in the deep of it being the pandemic, the hospitals at capacity, and a continuous rise in cases, and I am on my short daily alone time, aka walk home from work, I'll keep it short. He's a physician, by the way, I should add. I usually try to imagine having the patient in front of me with no intent or books around and see where my thoughts take me. So this is what I've got. With regards to epidemiology and the signs and symptoms, I'd say ocular toxocariasis. You might find serological positivity from Toxocaris species, although maybe not in the 60s, and signs of blood eosinophilia might be present. Differentials include loa, onchocerciasis, cysticercosis, but not supported by travel history or contact with pigs. With warm regards and admiration, AK. And he's an ID physician.
Michelle writes, hello, the wind is howling like crazy here in LA. It's a great day to stay in and enjoy the info you all share. You are my favorite edutainment. Thank you for sharing your journal clubs and case studies. My thoughts on 190. Here we evaluate a gentleman whose risk factors include rural location in North Carolina and contact with many dogs. I'm just a nurse, but I've heard that the saying, if there is galloping, think of horses, not unicorns. My guess is that a roundworm found a home in this man's eye, causing intermittent blurred vision. If I'm wildly off, don't worry. I'm listening and learning. All the best, and cheers to vaccine rollout. Be safe and well, friends. Michelle. I think it's think of horses, isn't it? Think horses, not
zebras.
Yeah. Okay. Sorry.
It is zebras. Yeah.
I prefer the unicorns. I'm going with that from now on.
I do too.
Wyatt writes, hello, TWiP doctors. I am sad to say that I missed out on the last case presented as we were finishing up our heme immunoblock this past month at Loma Linda and regretfully kept forgetting. I am thrilled to be back at it again. I had a quick question about the diploma in tropical medicine and hygiene. Who is the target audience of this program? Is it primarily for physicians meaning to refine their skills, PhD students looking to research tropical diseases full-time, anyone who has taken an interest, or all of the above? It sounds really interesting and like a lot of fun. It seems that it is to receive certification in the membership of the Royal Colleges of Physicians of the United Kingdom, but I'm not entirely sure how that translates to the healthcare certification of the United States. If you all know off the top of your head, that would be wonderful. I'll jump in with an answer to that at the end there, and Dickson as well, I guess. Anyway, on to my guess. I believe that the guilty parasite this month is Toxocara canis, which is a helminth that can be found in North America. Infection starts when the patient consumed the eggs of the parasite, which originally came from the intestinal tract and feces of a dog. The egg hatches in the small intestine of the human and then punctures the small intestine and migrates out. This is now classified as larva migrans and is typical of many different parasites. Because this parasite was able to move to the posterior eye and even lodge itself in the optic nerve, I am assuming that it is one of the visceral larvae that can migrate as opposed to the cutaneous ones. Now, the question pertains to where the parasite typically migrates to. Surprisingly, at least to me, these parasites can have typical spots of translocation. Some travel to the meninges, some to the stomach wall, some to the muscles, some to the lung and heart, and some to the eyes. Toxocara canis and cati are two that commonly go to the eyes. I arrived at this conclusion because I know that the parasite must be large enough to visualize with an ophthalmoscope, located in the eye and most likely from the dogs. These qualifiers ruled out Toxocara cati, the cat version of the parasite. There was one other parasite that caught my eye and that I had never thought of before. Thalassia callipata and californiensis. These cause a primarily cutaneous infection of the eye, and I think that they would have trouble moving to the posterior compartment. I also think that this parasite would be hard to find in North Dakota. T. calipadia is found mainly in Asia and Europe, and T. californiensis is found in the western U.S. and much less common. All this to say that T. canis is my final guess, and hopefully the poor chap is able to be treated. I'm guessing that they put him on albendazole in order to kill off any of the other worms. However, the eye may not respond to the drug regimen. It seems that surgery may have been the best option, but was no longer feasible at this time. All the best, Wyatt.
Joel and I'm sorry, who goes next?
You go ahead. Sorry.
Yeah, I thought he did. That's okay. Joel and Browen write, "Good day, TWiPeroos. It's Joel and Browen here from Brisbane, Australia. Today was cool and sunny, 32 degrees C, with the humidity dialed to an oppressive 93%. We'll try to keep this one short and sweet. Sounds like a case of ocular larva migraines. Our chief suspect for this presentation is Toxocara canis, a dog parasite endemic in the States. According to Otranto and Eberhardt's excellent paper, Zoonotic Helminths Affecting the Human Eye, the larvae of T. canis are the most common nematode infection of the human eye. They say that children are the most classically affected demographically, as was the case with a 3-year-old girl in TWiP 170. But this guy's beagle breeding is a bit of an occupational hazard. Dickson told us way back in TWiP 21 that T. canis stymied efforts to produce germ-free beagles. That's true. I still stick to that story, by the way. We were a little confused by the mention of surgical management. Up-to-date recommendations, albendazole. Otranto and Eberhardt say photocoagulation therapy can be indicated in cases such as this, where the larvae can be visualized and surgery is reserved for severe cases. But this treatment discrepancy may be a reflection of the vintage of this case. Albendazole does not appear to have been in common use in the 60s. Cheers, everyone. We love your work.
All right. Katie Jane writes, "Dear all, I'm writing regarding Dickson's ocular anomaly. The patient raised hunting dogs, so I decided my best bet was to look at zoonotic infections. I found a great article that greatly helped me out and provides a link. I've narrowed it down to Toxocara canis or Dirofilaria imitans. The larval form of T. canis can affect the eye, known as ocular larva migrans. It is the most common nematode infection of the human eye. The age range is possible for this stage of T. canis infection, and OLM may be caused by few or even one infected larvae. However, it seems that once the larvae are in the eye, they are there to stay, and the damage caused gets progressively worse. That was just my understanding. I might be wrong. Dirofilaria imidans is more commonly known as dog heartworm, for which we hopefully give our beloved companions a tasty oral treatment each month. This parasite is prevalent across the U.S., but particularly in the southeast. According to the article linked above, this species of Dirofilaria has been reported in the anterior chamber of the eye. Furthermore, Dirofilaria species worms are often motile and noticed by the patient because of interference with vision. Removal is curative, and full visual acuity is generally restored with no long-term sequelae. I think this is the one I'm going to choose for my guess. I found this one a little tricky, and I'm not 100% convinced by my choice of answer, so I'm looking forward to hearing other people's guesses and the resolution of this case. Thanks for all you do. Wishing you a wonderful start to 2021 and another year filled with science education. Oh, wait, I forgot to mention the weather. 41 degrees in northern Wisconsin, well above average for this time of year, and no complaints here. Katie Jane is a faculty in North Central Technical College in Wisconsin. Daniel.
All right. Martha writes, Dear TWiP, I'm a bit late in responding to your latest puzzler regarding the dog breeder in North Carolina in the 1960s with an eye parasite. I was reminded to write while listening to Twin. I must admit that most of that podcast goes over my head, but I was struck by a comment, Dr. Calipari, that something can be aversive without being painful. For me, although I am not usually squeamish, looking at eyeballs infected with parasites is aversive. So I delayed responding. I'm not familiar with eye parasites. I went looking for images of the eye to acquaint myself with the anatomy, and then onto images of parasites infesting eyes, including those open to better demonstrate the parasite in situ. I will be brief because my search was rather superficial. I kept closing my eyes. I considered toxic keratoconus, which can infest the human eye, but it is said to present as a granuloma on the retina. Thalassia calipeda seems to specialize on infecting the surface of the eye, resulting in some nightmarish images. However, not what Dr. Despommier described. So I'm guessing it was Henia crassips, which can cause ocular loss of migrants in humans, and which Wikipedia assures me adult human immune systems typically will remove before permanent damage is done. Thank you for this and all the TWi podcasts. You may wonder that someone who is too squeamish to look at infested eyeballs would want a book of parasitology. I'm going to desensitize myself by watching La Chienne Andalou. All the best wishes, Martha.
James writes, through a glass darkly. Really think it's visceral larva migrans from the hunting dogs. Loa Loa came to mind, but I don't recall it going so deep. That's one I'm actually diagnosed on a blood film in an African immigrant who gave the history of a torpedo swimming across his visual field. There's lots of protozoa, acanthamoeba, toxoplasma, malaria, that can cause various visual symptoms, but I really doubt Dickson could see those in his scope. And you're right, I didn't see those in my scope.
Drew T writes, Hi, dear TWiP team. My differential for the last episode is as per geographical distribution. Most likely, 1, angiostrongyliosis by Angiostrongylus cantonensis. 2, Baylisascariasis by Baylisascaris procyonis. 3, trichinosis by Triconella spiralis. 4, onchocerciasis by Onchocerca volvulus. 5, toxocariasis by Toxocara canis and cat eye. 6, dirofilaria by Dirofilaria repens imidis. Least likely, loiasis by Loa Loa, and Bancroftian and Brugian filariasis by Wucheria Bancrofti and Brugia malayi. Okay.
That was impressive. I feel like my ability to pronounce things has diminished as Vincent's has grown.
Well, some of us learn, and no.
I'm going backwards. We're like passing each other in time. Vincent is becoming the expert parasitologist, and my knowledge is being sucked out. Kia ora from Tongaroa from Andrew. COVID, New Zealand is buying enough vaccines to vaccinate everyone in the country and more to give to our Pacific Island neighbors. Proud to be a Kiwi. Weather, an unseasonably cold holiday, 15C and rain. Actually, I should say, Andrew, we've bought enough too, and we've got them stored in locked freezers. So don't you worry. We've got plenty of vaccines here too. Case 138. My guess is ocular larva migrans caused by Toxocara canis. This is similar to case 138, which was Bailey ascaris from raccoons. If the dogs the man raised were hunting raccoons, this is also a possibility, as the eggs might be transmitted by the fur coming in contact with raccoon scat.
Come on, Dan, you got to pronounce that.
I know. This is, didn't I just mention this? So it's Ne Mihi. Did I do that right?
Sounds good to me. Na Mihi. Dickson, I think you're next, right?
Yep. All right, I am. Erica writes, hi, it's 5 degrees C and overcast with possible flurries in Toronto. I started listening to TWiV earlier in the year for obvious reasons, and TWiP more recently, because parasites are equal parts fascinating and horrifying. I think it's awesome what you're doing for science communication, and it's been great having your extensive archive and regular updates to keep me company throughout our various stages of lockdown. Also, I grew up in Maplewood, and it's nice to hear something good coming out of New Jersey. That's not too far away from Fort Lee. There's more. Keep listening. So with the disclaimer that I have zero relevant training or experience, I'd like to submit my guess for the case study, Bailey ascaris infection. Hoping for a book and wishing everyone a less bonkers 2021.
And Maplewood's very close to me, Dickson.
Yeah, it's not too far.
All right, this is from Chelsea on behalf of the University of Central Lancashire Parasitology Club. Writes, happy holidays, TWiP professors. It's another cold and wet day here at the University of Central Lancashire, but we've been busy trying to solve this case over the Christmas period. We have a number of contenders, though the likely culprit in this case is ocular toxocariasis. Ocular toxocariasis is most likely caused by the ubiquitous Toxocara canis. As the case states, the patient works with dogs and other animals which are common hosts for the pathogen. It's likely the patient became infected with Toxocara when they unintentionally ingested fecal eggs or larvae via poor hygiene practices, for example. The larvae then migrated into the eye. Once within the eye, its clinical presentations can be classified into one of 4 forms: posterior pole granuloma, peripheral granuloma, nematode endophthalmitis, and atypical endophthalmitis. Huh? You want to make an apoptosis, don't you? Okay, endophthalmitis and atypical presentations. We believe the patient may have suffered from a peripheral granuloma with atypical presentations of optic nerve swelling and other anterior findings, cataract, conjunctivitis. In some cases, a biopsy of the infected tissue can be used to diagnose OT. However, this process is risky and difficult in the eyes. Thus, typical diagnosis involves identifying the clinical presentations of a local granuloma in the retinal posterior pole or periphery. Serological tests can also confirm diagnosis and may include enzyme-linked immunosorbent assay for detection of the serum antibody against Toxocara larvae. Our second contender for this case is anterior ophthalmomyiasis caused by dipteran flies.
Beautifully pronounced. That was
like OT.
I have
the
to
larvae
pause
may.
here. You were fantastic.
What's that, Dickson?
I said I have to pause here. That was an amazing pronunciation.
It's okay. It's no big deal.
It was perfect. No, it was perfect.
Thank you, Dickson. It's very kind of you.
I try to be kind.
Like OT, the larvae can migrate into the eye upon ingestion, but is less common in infected patients and clinically appears as anterior uveitis. Uveitis, Daniel? Uveitis, right?
I think uveitis works for us here.
Uveitis?
Uveitis.
I was asked to include some eye puns, but the jokes couldn't be any cornea, so I decided against it. Oh.
Decide against it.
Yeah, well, you just read the next one.
And Happy New Year, Chelsea, on behalf of the University of Central Lancashire Parasitology Club. Very cool. Oh, and
That's
Daniel,
very
you
funny.
get the real
You
Kevin.
get the good one. You get the good one.
Okay, the other Kevin. Kevin writes, Dr. Despommier, like some film noir gumshoe, had one in the chamber, but the worm got away, lost to follow-up, as the saying goes. There's nothing funny about this aqueous humor. The whole case is going to the dogs. Last of the year, therefore a sense of urgency. Research results in this case are a throwback to the era of anecdoting case reports. Controlled trials are near impossible in these extremely uncommon cases. It's a refreshing change to read a narrative uncluttered by references to interleukin 23, chemokine receptors, and whole genome sequencing. Our 50-year-old dog breeder has not traveled abroad, but I will not let that fact prevent a discursive excursion. Before a quick differential, I highly recommend the comprehensive Table 1 in Otranto's 2011 review. Since our patient hasn't traveled, the tropical filariases can be ruled out. These usually involve the subjunctiva, and anterior chamber involvement is uncommon. Likewise, with gnathostoma acquired by raw fish eating or copepod ingestion, and usually seen in Asia, etc., many other agents that have been the subject of case reports can probably be eliminated. Angiostrongylus, enterobius, dipetalonema, various trematodes, cestodes, and cysticerci, pentastomids, and maggots, all referenced below, leading contenders for Dr. Despommier's elusive worm in descending order of my estimated likelihood. Dirofilaria, which we know as heartworm, many species, two relevant here, D. repens and D. tenuis. Larvae are approximately 20 microns long. They've been described in the anterior segment, but are usually subconjunctival. Baliascaris review mentions getting infected via exposure to raccoon latrines. No raccoon latrine is not the name of a punk band. Raccoons do not defecate promiscuously. Rather, they deposit their droppings at designated sites. Our dog breeder may have stepped in it, but infections are uncommon. CDC reported 11 total USA cases in a 16-year period. Note, however, that the CDC states that dogs can be the definitive host for Baliascaris and shed eggs, which could have been ingested by our patient. This nematode larvae measures up to 1.8 millimeters in length and can cause ocular, larva, migrans, encephalitis, and other serious issues. Toxocara. Otranto states that Toxocara species are probably the most common cause of nematode infection of the eye. Our patient's intimate connection with hounds makes Toxocara the likeliest candidate for his anterior chamber findings. The larvae measure 400 by 20 microns. They can be killed by photocoagulation. Serologies are often negative in cases of ocular larval migrans. Treatment usually involves albendazole and corticosteroids. Final note on treatment, the title sums it up. Eyeworm swimming live in anterior chamber. How to remove live nematode without damaging eye structure. Irma Suwandi Sadakin, simply forceps and lidocaine. Various other methods have been reported. Some involve paralyzing the worm with lidocaine or related drugs, use of simple irrigation aspiration, and the installation of viscoelastic substances, e.g. methocellulose, that gums up the worm for easier extraction. Not a do-it-yourself enterprise. Happy New Year. And then we have some end notes. And let me see. We got a lot of end notes here.
Go down to the terminal and just read that one paragraph of the terminal curiosity. That's interesting.
Okay. I'm jumping. Digressive to the point of pointlessness. But this mental pathway was stimulated by the known, especially by Dr. Despommier, phenomenon of frog meat and snake meat poultices and the association with ocular sparginosis, mercifully omitted from the above discussion and tediously expounded upon in TWiP's 169's terminal curiosity. The subject of poultices brought to mind the whole gamut of devices touched upon in TWiP 189, talk of cholera belts and such, to wit, waistcoats, girdles, belts, stomachers, cummerbunds, wraps, compresses, plasters, poultices, and fomentations. I suggest reviewing Dr. Despommiers on frog poultices for the Columbia University Online magazine, and we get a link there.
Dickson, did you know you wrote that article?
No comment. That's not even a question. Come on, of course I knew I wrote the article.
About the Chinese episode, the sort of story about the father and the son with the eye issue. And
that's part of that story. That's part of that story. Yes.
What's the name of that story? What did you call that story, Dickson?
Well, that I'd have to go back and look up. I'm sorry.
Are you serious? You don't know what it is?
Tell me how many articles you've written that you can remember the exact titles of.
I can remember all of them.
I'm sure you can.
I'm impressed. I don't know if I could, most of my titles, right? Because now we write a title where it's your entire conclusion in
exactly right.
That is the maximum word limit. So
at 3 a.m, Vincent, I'll call you because I'll remember it by then.
All right, I'm going to read it. Do the next one.
Elise writes.
Elise writes, Dear TWiP Trifecta, I'm writing to you on New Year's Eve from Lower Manhattan and wishing you all a better 2021 than the year that is finally departing. It is a dampish 39 degrees Fahrenheit, 3.9 degrees centigrade here at the bottom of the island. I'm not confident in this diagnosis, but after missing the previous opportunity, I'm determined to submit one. It was hard for me to sort out which sort of parasite Dickson saw in the patient's eye, but given that it was causing intermittent blurred vision in the evenings, I suspected it was a helminth. Dickson was very gleeful about what a sight it was, so I know it was easily visible, and worms in the body always seem to be more lively at night. A few parasites do show up in human eyes, and I suspect that the infection might be toxocariasis, given that the patient ran a kennel and had a lot of exposure to a lot of dogs. My diagnostic lack of confidence comes from the fact that I read that ocular toxocariasis can damage the retina, and if this gentleman ever got it removed, perhaps the outcome was worse than was implied. The recommendation would have been to remove the worm, but this one wandered off, and I don't know if it would cause more problems. Well, I did find other discussions of helminths that show up in human eyes. Toxicara seemed to be the most likely given the lone symptom. But as I said, I'm not entirely confident. As always, many best wishes to all of you. Thank you for all of your podcasts and all of your work that keeps me engaged, distracted from less pleasant thoughts, and utterly interested. All the best and Happy New Year. It's very nice. P.S. I know this is a crossover reference, but Vincent, I was on the Upper East Side this week and I thought of TWiV because of this site that you mentioned with some nostalgia.
Yeah.
I'm scrolling to the next one.
Is that the Papaya King by chance?
Yeah.
It is.
A photo of Papaya King, which I've talked about on TWiV. That's lovely. At least when the pandemic is over, I'll meet you there for a juice sometime.
There you go.
Cool. Dickson, the name of your tale was Frog Legs and Parasite Tales.
Yes, it was.
It's just a Google away, Dickson.
Well, I'm just a Google away.
Indeed you are. All right, let's go to the next one, which is Josie, Dear Triumphant TWiP Master Trio. It seems that writing answers to your parasitic puzzles is becoming the way I pass my time while biology takes over my life. Last time I wrote in, I was recovering from oral surgery, blamed my bad answer on the codeine, and now I'm writing while quarantined, waiting out my time while my essential worker husband gets a 14-day break from his essential work. Good thing I like medical science since I am using so much of it lately. As far as Dr. Despommier's mystery case goes, it's very clear to me that the man with the blurry vision has become a dead-end host for a bookworm.
Very good, very good, very good.
Normally devouring any available reading material, this poor creature must have been munching happily away at the evening newspaper. The unwitting host also enjoyed devouring, was accidentally consumed, and migrating through various organs ultimately settled in the host's eye because there it could see out and continue reading whenever the man himself also sat down to read. This inadvertently causing visual distortion for the host as the view was now somewhat blocked. Seriously, though, I seem to remember that the man raised hunting dogs, implying contact with raccoons either because they are the intended prey or because they are helping themselves from the dog's dinner. Could the mystery worm have been Balis ascaris, contracted by contact with raccoon feces or whatever surface the feces was deposited in a week or two before? Weather here in Library Hell is currently comfortably chilly, 39 degrees F, 4 Celsius, 3 degrees Reaumur. For the occasional complainers that have grumbled about your weather reports over the years, they should know better. All good scientists take note of their field conditions and record them as part of their work. That's great. I love that. Happy vaccinated New Year, Josie in Library Hell. Lovely.
All right. Leland writes, hope this gets in time. Ocular larva migrans, Toxocara canis.
Wow.
That was
quick.
a short one. You want to read the next one too?
I'll go for it. I'm feeling like I got the energy. Katon writes, Happy New Year, TWiPers. It is a beautiful sunny winter day with a winter blanket of snow here in the Upper Peninsula of Michigan. Beautiful spot, by the way. The icicles are melting this mid-morning and the thermometer reads 40 degrees. Although I am just a country farmer, I still would like to give a guess of the identity of the entity that haunted a beagle breeder's eye in the 1960s. First, I would like to point out that as Dickson presented this case study, he said that he saw the mystery entity with his own eyes and said he was 95% certain of what it was. Yet he gave us no description of size, shape, or color of the organism. I believe he omitted that information because revealing it would make this exercise way too easy. I believe Dickson saw some sort of zoonotic roundworm that had migrated to the gentleman's eye. I am 95% sure it was the canine species Toxocara canis because about 30% of dogs younger than 6 months deposit Toxocara eggs in their feces. Other animals have their own special species of roundworms, but they can also find their way to the human eye, such as cats, Toxocara cati, raccoons, Bali ascaris, procyanus. Both could have been possible candidates for the gentleman's infection in his southern rural environment. Lastly, if the gentleman had traveled abroad, say to Africa, he could have gotten loiasis, also known as the African eyeworm. It is caused by the parasitic worm loa loa and is passed on to humans through the repeated bites of deer flies, also known as mango flies or mangrove flies of the genus Chrysops. The loa loa worm most commonly migrates to the human eye, but can also invade other parts of the body. The following video shows the removal of a loa loa worm in the eye. It is both fascinating and educational. We get a link. I have to agree with Dickson. The study of parasites is endlessly amazing.
Nice.
Vicky writes, hello, beloved TWiPers. It is 19 degrees F in Duluth, sunny, but a chance of snow tomorrow. I received my used copy of the second edition of Parasitic Diseases last week, a collector's item, by the way. On the first page of the table of contents, someone had written, no more lettuce. That wasn't my copy. And I hope that advice is no longer relevant. I was relieved to see that the images are black and white, as vivid images could give me nightmares. I'm guessing that this poor fellow had ocular Toxocara migrans, not as uncommon as I would wish. People going to see an ophthalmologist for vision problems have this at a rate of between 0.1 and 1% in warmer climates. The American Journal of Tropical Medicine and Hygiene calls this a neglected disease, sore neglected. I'm pretty sure that I read somewhere that Toxocara larvae don't migrate to the optic nerve. So does this indicate that my guess is wrong, or that this migration is new to science? What other larval parasites migrate to the eye? As far as treatment, albendazole or mebendazole are recommended, but not FDA approved as of 2014, and steroids for inflammation. I'm worried about this fellow's vision and anxiously await your podcast. My dear friend Jean, when she was a child 60 years ago near the Canadian border, used her precious few coins to go to the feed store and buy deworming medicine for the barn cats and dogs. It sounds as though she would see dead worms in their feces. She thinks that she has developed a parasite phobia, but I don't see how that's possible. Negative feelings about parasites are normal. She grew up to run the chemistry lab at one of our local hospitals. Coincidence? Yours in parasitology, Vicky. Yeah.
Alex writes, Dear professors, just after listening to your latest episode in which Professor Despommier presents a case of a patient with an ocular parasite, my podcast app presented me with an older episode, namely number 40, which is aptly titled, Doctor, There's a Worm in My Eye. This, of course, made the diagnosis a lot easier for me. The patient most probably suffered from a case of ocular larva migrans syndrome from Toxocara cani. As a dog breeder, exposure is quite clear in this case. Differentials include loiasis, onchocercosis, cysticercosis, and gnathostomiasis, but all those nasty buggers are very rare in the U.S. That said, I must say that I envy you for having seen this fascinating creature in the flesh. Such an occurrence should be enough to turn anyone into a passionate parasitologist. Take care, and all the best, Alexander, 3C, 38F, and Cloudy in Vienna. Nice.
Right.
All right, Kate writes, hello, this is a guest for episode 190, The Man with the Blur in His Vision. I believe he had ocular larva migrans caused by Toxocara canis. I'm writing to you from the parking lot of our pediatrician waiting for my daughter's appointment, but I didn't want to forget to write in. Maybe I can win shortest guess, second shortest guess. Thanks, Kate from Knoxville, Tennessee.
Great.
And I'll finish off Owen's short one here. Wayne. Dear TWiP, to my shame, I didn't submit a case guess last month. Apologies for the absence. My guess for this month's case is a worm of the genus Toxocara, most likely Toxocara canis, given the known dog exposure. I don't know of many other worms that get into the eye as described, although Tinea, Onchocerca, and Loa Loa all cause significantly different kinds of eye infections. If you have time, a brief overview of eye worm infections from you experts would be great. A brief question for Vincent. I'm using the dedicated Microbe World app to listen to the podcast. It works very well, but sadly, I can't see your newer podcasts like TWiBo and Immune on there. Is this just because I'm doing something wrong from a technology perspective, or are they not uploaded onto the app? I'm not as good at technology as I am at eyeworms. I hope you're all well. Yours sincerely.
That's funny.
They're not there, Owen. The ASM has actually stopped supporting the app, and as I made the newer podcast, they didn't add them on, and I don't think they're interested in doing it, sadly. All right, there we go. 23 guesses, plus a couple that already won a book, so that's good. Nice job, folks.
Right.
I think we have two more people to guess, right, Dickson? Two more people get to guess.
I would think at least two more people, perhaps a third person.
Vincent, what were your thoughts? Should we let you jump in on this?
Yeah, so my differential included loiasis, gnathostomiasis, onchocerciasis, and toxocariasis, and I figured the first three are pretty rare in the U.S., so I would say toxocariasis, like many listeners have. Dickson, that you know.
No, that was my case.
Well, you don't get to guess. I don't get to guess. Yeah, no. So I remember having a very similar case to this presented to me when I was in Tamil Nadu. And they were sharing a case, and they described, much like you described, and then they said so. And and they asked around, what do people think it is? And what people responded? Ocular parasitosis. And then they moved forward. I was like, wait. I mean, we knew that.
We narrowed it down to 150 entities.
It's very funny.
So I would say, it would be great to have seen it, right? And I think as some of our listeners suggested, what was the size? What did it look like? Little subtleties like that. But, to sort of go from ocular parasitosis down to ocular larva migrans, which you could say, Dan, that's not even much more specific. You're talking about a parasite larvae that migrated to the eye. But then to be even more specific, my sort of highest in the list of most likely would be the Toxocara canis. But I think that, there's a slightly larger differential, but that would be my sort of top pick. And then sort of, hopefully based upon size and what Dickson actually saw.
Right. So I'll be frank and honest with both of you, and in fact with all of our TWiP listeners. In in telling you that we never did find out exactly what it was because it escaped before we had a chance to sample the goods. However, let's just use some logical thinking here and think about the size of the worm. And it was a nematode. It was definitely a nematode. And it was doing, I'll do it with my hand because I think most people can see this on YouTube. It's going to go sort of like, it has serpent-like motion. My hand is not exactly coming.
Oh, now it's actually on the screen.
That's right. It waves back and forth like a worm or like a snake trying to make its way through water or through grass. Okay, so it's going
serpentinous,
rhythmically back and forth. The point is, this worm has to be large enough to cause blurring of this man's vision. And if you'll read the fine notes on the chapter on toxocariasis, the larvae are the only stage that humans can acquire. They don't get the adult worms. So larvae are microscopic. So you can't see those with an ophthalmoscope. They're unavailable. And besides, even if you could see them, they would be so small that they couldn't possibly cause blurred vision in this gentleman's eye. So our best guess was Dirofilaria, but not the larva of Dirofilaria, an immature adult. The worm was clearly the width, or the length, I should say, of the diameter of the iris. So that's pretty big. To see that with an ophthalmoscope, you could probably, with a hand lens, you could probably see it also. The point is that when you saw it in the ophthalmoscope, you thought you were looking at a gigantic serpent swimming in an ocean of Atrius humor. It was fantastic to see this because everybody was excited, including the patient. He wanted to see it himself, and he said, "What are you looking at?" And we said, "We're seeing your entity. Or do you have blurred vision right now?" And the guy says, "Yeah, I can barely see out of my right eye." So the point is that the Toxocara cannot do that. The larvae are too small. The other worms are too exotic to be found in rural North Carolina. So the only logical choice, and some of our guesses were right on the money, they guessed that it was Dirofilaria. But they didn't guess that it was Dirofilaria adult, immature, because they do grow up to be immature adults. And in fact, some of them can actually be found in lung tissue. Some of them can actually be found in the anterior chamber of the heart. But they, what happened to this worm, we don't know, because it eventually ended up in this man's gray matter, presumably. And it caused no further harm. It didn't do anything. And in fact, when it ends up in the lung, it ends up as a coil of worm, and then it calcifies. And so they call it a coin lesion. Coin lesions, Daniel, right, are associated with other entities like lung cancer, for instance, and some other things.
Yeah. I like the way you're thinking this through because the size is an issue, right?
But the guy had blurred vision. That's the clue. That was the big clue.
Yeah, because the Dirofilaria they can get so big, right? An adult can actually get to be about, we'll say 20 centimeters, right?
Exactly.
But they're not like, and so when you see them, so often someone will have an abnormal chest x-ray, they'll have a pulmonary nodule, they'll have a nodule in the lung. And when you actually biopsy that nodule thinking, oh my gosh, this is cancer, there is a 20 centimeters, so about a 9-inch, 8 or 9 inch worm that's curled up in a little ball. And that's what's pulling out of there. So that makes sense. Some of the people brought up Bailey ascaris, right? One of the things about they can also continue to grow. That's one of the concerns that they get into the brain tissue, they can grow.
Sure.
But that, I think the man would have come back if it was that, right? Because this
is true.
Where this will go off somewhere, calcify, that'll be the end of it.
That's right. Even if Bailey ascaris larvae do grow, and of course they do, once they get into the brain, you take a look at their size, they're about twice as large as Toxocara canis or cati larvae. Now, those larvae are still microscopic. I mean, there's 60, 70 microns in length. The Bailey ascaris might be 120, 140 microns in length, but that's not visible. That would never cause blurring vision in a gentleman sitting in the evening reading his newspaper and sipping his bourbon. So I think that just presumptive evidence suggests very strongly the only real guesses that make any sense with regards to the epidemiology of this is a immature adult of Dirofilaria. That's a lot of people are going to be very unhappy with me because
no. You know what? I'm gonna concur with you, because if you were able to see this and it was large enough,
it was big.
This makes a lot of sense.
It was big. Now, you've seen it on ophthalmoscope.
But how did you rule out the bookworm? Because I really like that idea. That it was a bookworm.
No, I think those are the two choices that I would go with. And in fact, speaking of books.
Excellent. Let's give away a book.
Let's give away a book.
All right, we had 23 guesses. We need a random number between 1 and 23. Right. Are you ready,
You want
Dickson?
a drum roll? I am.
The winner is number 17, and that is Josie, or Yossi, winner. So there you go, Josie. And please send your address to TWiP@microbe.tv. Josie in Library Hell. I don't know if I can mail it to Library Hell, but we'll try.
You might need to just write that, and then everyone knows where that is.
That's right.
All right, so we have Daniel has a hard out at 5:30, so let's spend 15 minutes or 20 minutes on this paper. Dickson, how's that?
I would love that. We can summarize briefly by starting with the title. So this is an article that was written in the journal Science. The first author is Wendt, and the last author is Collins, and there are some 6 or 7 people in between. It's co-authored by Zhao, and the title is A Single-Cell RNA-seq Atlas of Schistosoma mansoni Identifies a Key Regulator of Blood Feeding. This is a fabulous article, but let me just state that before this article was published, there was a write-up of the 2 articles that appeared together. This was the second article, and I picked that because I didn't think we had time for 2 articles. So there's another article that precedes it that also has a unique approach to studying the biology of schistosomes. And I would like to read the last paragraph, or last sentence, I guess, in the review of both of these articles that appears in the beginning of Science, because it's quite telling. Wang et al., that's the first author, and Wendt et al., that's the second paper, go much further than identifying drug targets, supporting the premise that it is critical to understand the basic biology of pathogens before designing strategies to kill them. Vincent, do you like that sentence?
Basic biology, baby. Love it.
Do you like that sentence? I love that sentence.
So true.
So it's very tough to convince somebody that what you're going to discover in the lab someday may save the world, but it may very well indeed be true. So this article basically says that we took adult male and adult female Schistosoma mansoni worms, very large worms. In fact, you don't need an ophthalmoscope to see these. They're big enough to see with the naked eye. They live as pairs of worms in the portal circulation of the small intestine that leads back to the liver. They feed on your blood. But how they feed is subject to a lot of investigations, and what do they feed on, and can we interfere with that process? At the same time, they're multicellular organisms that have a nervous system, they have an excretory system, they have muscles, they have a surface-coated tegument, as it's called. It's not a hard shell like you'd expect in a nematode. It's soft, pliable, and as it turns out, it's important for its acquisition of nutrients by simple transport across that membrane that cuts off the parasite from the host. But most importantly, it eats blood. And it eats blood, digests the red cells, takes out the globin part, and culls out the amino acids using an enzyme called cathepsin B. And then it's stuck with all this waste. The stroma of the cells, for instance, the heme portion of hemoglobin. And so what it does is it then regurgitates all of that stuff back into the host's bloodstream from whence it came. It has a very interesting mechanism for clearing its gut tract. When it ingests blood, of course, it contains a lot of other formed elements besides red cells. It contains white cells also. And it has a special mechanism for sequestering the white cells up in the front part of its gut tract to keep it out of harm's way while it digests red blood cells. It's an extremely sophisticated organism. The authors then set about finding out what the transcripts were for each individual cell type in this worm. They sequenced the RNA constructs from 43,642 cells. In other words, they completely knocked the worm apart into individual cells, cell-sorted them, and then subjected each individual cell to an RNA-seq analysis to show what it was producing, to try to identify where that cell came from. They identified 68 different cell types, including specialized stem cells that maintain the parasite's blood-digesting gut. They made a lot of discoveries along the way. They discovered where the flame cells come from, the excretory cells. They learned a lot about muscle cells. They learned a lot about the tegumental cells that coat the worm. But most importantly, they stumbled upon a molecule that they were producing called HNF4, which stands for hepatocyte nuclear factor number 4, which turns out to be a transcription factor. Now, we have that same protein in our repertoire, and I looked it up, of course, to find out what it did. And it has multiple functions, and it regulates the production of various things, depending on what you are. If you're a human being, it does certain things, and if you're a worm, in this case, it regulates part of the developmental cycle of the development of the gut tract of the worm. So what they did, I'm cutting to the chase here because our time is limited. They developed RNAi in constructs for this gene, and gene product, I should say, and then used that as a way of interfering with the developmental process, and then implanted these altered adult worms into mice to see whether or not they could produce eggs. These worms are highly manipulable, and you can play with them in vitro and then put them back in vivo and find out what you've done to them, whether that has an effect on their life cycle or not. In this case, interference with this particular gene product, HNF4, transcription factor, interferes with the ability of this worm to maintain itself in the host itself. They had a 60% mortality rate of worms that had this gene product altered by interference, which suggested to them very strongly that this is a target for a better drug than the RNA that they were using to try to completely ablate the effects of this one product. But searching through this morass of 43,642 cells, they produced an amazing body of work, which now is a catalog of schistosome cells for lots of other people to make use of. Their diagrams are easy to see, easy to read, easy to understand. The work is complicated, it's involved, but the expression of that data in the form of various drawings next to the actual event, the picture of the worm itself, made it very easy to follow this result all the way to the finish line. There are lots of pages to this article. This is not a common article length for Science magazine. They usually cut them off much shorter than this. This article deserved a lot of attention, and that's what it got. I would say that on the whole, there has not been another paper or another investigation on schistosomes which has been so global, that has identified so much new work that could grow out of this one particular approach. And I'm sure that it was an idea whose time had to wait for the proper technology to come along so that you could answer the question that you asked a long time ago, and that namely, what do each of these cell types in this worm do? How do they do it? What do they produce? And can we somehow interfere with the biology of this worm by learning more about their normal biology and create an abnormal state in them by using either drugs or antibodies, et cetera? So I'm absolutely stunned by the ease in which I understood this article, because I'm not a trained molecular biologist by any means. I mean, I understand what a transcription factor is and all these other things, but to see all of this being applied to an entity that I have known about since I was a graduate student. In fact, we played with this life cycle while I was here at Columbia University as a graduate student. We had access to it. We had some snails. We had the worms. We obviously didn't have a clue what to do with them. These people have all of those tools plus all of these new tools. And now being able to ask the right questions, you can get the right answers. So I think if we continue to see articles like this written about multicellular parasites in the future, there is a bright light ahead for possible interference with these parasitic infections using technologies that we're just now coming to grips with. How did you look at that, either one of you guys?
I'll jump in, Vincent. I mean, I have to say, like, you start to read it, and I read it a couple times. It sort of gave me pause. I was like, "You did what?" So,
exactly.
Right? They took they took a schistosome, and they basically broke it down into single cells, right? And then put those single cells in different wells, and then did a full RNA-seq. Of a single cell. They did that enough times that they characterized 43,642 cells, right? I mean, I'm just thinking of how much that cost, right? That was not cheap.
No.
And then, I mean, this is like reductionist science, right? And then they sort of rebuilt the creature by looking at each of those different cell types, getting them to sort of cluster and group and saying, okay, now we've got 68 sort of clustered distinct cell populations based upon this RNA-seq data. Kind of amazing, actually. I have to say, I'm trying to think of how the RNA, stays the same, when you take an organism like this and you pulverize it into individual cells, right?
Yeah, that's
also
a good question.
Interesting, right? That you wouldn't say, okay, we're going to take cells from here and cells from here and cells in the gut tract and sequence those in mass. So it was very interesting, creative approach. But I think the thing, it gets to sort of the issue, we have basically one drug to treat schistosomiasis.
That's right. That's right.
And if we lose that drug, right, and many would argue that this is one of the, if not, one of the top parasites as far as causing morbidity, suffering, death throughout the world. Hundreds of millions suffer,
Indeed.
And we have one drug. Kind of crazy just to see. So I think it's critical that we do science like this because, one day we're going to lose that drug. One day that drug's not going to work so well. We're going to need other options. So I think this work is really critical.
Yep.
So there were two things I thought were of interest here. First, they say we uncovered unexpected molecular complexity within the schistosome nervous system.
Right, that's true. That is true.
They say this complexity is surprising given the relatively sedentary lifestyle of the parasites in the portal vasculature, right? So that's kind of interesting. And the other thing I wanted to mention was that if you this gene that Dickson focused on, HNF4, when you knock it down, it knocks down a protease, cathepsin B, which they believe is important for digesting the hemoglobin. So that's kind of nice, and it identifies at least one of the targets of this transcription factor that's important. So, I mean, it's nice that they did a functional experiment because they could have just published a catalog, right? Yeah. So at least now we see one gene they knock down and it has an effect. So hopefully people will follow up. And Dickson, if you had lived, if you had researched in this era, you'd be doing this with Triconella, no doubt.
Absolutely. No question about it. No question about it. I want to say one other thing about cathepsin B, and that there were earlier studies which sort of pointed in that direction where they isolated cathepsin B, the protein, and they injected it into mice, and they actually got some protection from that. And I reviewed that literature today just before we went on with our podcast to say that, to find that this one transcription factor actually regulates a portion of the gut tract that might synthesize that, which are these glands in the front of the esophagus, which secretes those proteins into the gut tract. It's a blind gut. That's how they digest their food. To see all this work come together, it's just wonderful. That's absolutely wonderful.
Dickson, how many cells are in a Triconella worm?
It's a heck of a question.
Are they bigger or smaller?
I know. No, they're much smaller than these worms.
Yeah.
Yeah, and the number of cells is fixed after the third stage larvae are produced. So
Okay.
I think there are a couple of hundred. It's like Caenorhabditis. It's a little bit like Caenorhabditis in that sense. It has some specialized cells, but there's not more than, let's say, 500 different cell types.
Easy piece of cake to do it with Triconella.
Well, 500 cells total, 500 cells.
Yeah.
I didn't mean cell types. I mean just cells. That's how many cells there are.
Well,
piece of cake, right? Piece of cake.
Very good. All right.
Right.
Let's see if Daniel has another case. I don't know.
Yes.
I was tossing over in my mind like several cases. Which one do I want to talk about? And I'm going to throw everyone a bone. I think this is an easy case. If I apologize if I say that and you don't get it right. But what maybe people have learned about me recently is now suddenly everyone wants to hear me talk about infections all the time. My family just knows that's what Dad does, right? The time that my youngest son Barnaby dressed up as a guinea worm, for show and tell, and he didn't have to look anything up. So this story comes from a few years back when I go to visit Malawi. And I go to the capital, a long way of Malawi, where my wife's sister's living, of course, right? This is the same story where I got up in the morning dressed in a suit and tie, and my kids are like, "Daddy, what are you doing? We're on vacation." I said, "Oh, yes, I'm going to treat HIV patients at the Central Hospital today." We're meeting a lot of people, and we're meeting a lot of people who work for USAID and people who work at the embassy. And I actually get to meet one of the clinicians that is taking care of a lot of the government people over there, as well as their children. It doesn't take them very long to realize what Dan likes to talk about on vacation. And he says, "Oh, I've got this great case. Let me tell you about it." For him, he felt like it was a very straightforward case, as I'm going to suggest. One of the sons of one of the USAID workers is asked to be seen by him. Because the son's urine has started to turn red. He's urinating blood. The parents are quite upset. They're actually new to Malawi. They've only been there for a short period of time. So we get a little bit more of the history. What's the story? And it sort of reminds me of one of the earlier emails is, the dad's excited. It's hot there. So they've been going on the weekends about 2.5 hours, I believe it's east from a long way, down this long road to where there's this beautiful lake. And there is a place where you can go swimming. It's they call it the Calendar Lake, Lake Malawi. There's a place where people can stay. They have these buffets. And it turns out the family has been swimming in Lake Malawi. So he says to the parents, "It's okay. There's no reason to panic here. I want your son to take 2 pills in the morning, 2 pills at night, and this will clear right up." And it goes ahead and clears right up. No diagnostic testing is done. The son gets better. What do we think is going on?
How old is this worker who's?
So it's a teenage son, and I should say new to Malawi, not that new to Malawi, because I think that's important. They didn't just get off the boat. They've been there for a little while. Maybe I think it's more proper to say new to sub-Saharan Africa.
And the rest of the family is okay.
We can have a discussion about that as well, because the rest of the family has been swimming in Lake Malawi as well. And perhaps they have not been vigorously toweling off when they get out of the water. We'll throw that as a hint as well.
That's what Daniel did once. He vigorously toweled off, right?
Or forthcoming about the color of the urine.
Yes.
So any other medical issues that we should know about?
No, everyone actually in the family is quite healthy. That's one of the things. These posts where people go, where people have to be healthy in general. You don't want to send people with significant medical problems, because should you have an issue, you're going to have to do a medevac to Johannesburg.
And they have all been swimming in the lake, including
they've all been swimming in the lake, and it's just the son that reports the red urine.
And then someone said take 2 pills in the morning and 2 in the evening for how many days?
That was it. Just one day. And miraculously, things get better.
Oh,
Miraculously.
excellent. All right. Okay. Anything else, Dickson?
Nope. You've asked all the relevant questions.
Dickson, you don't have a hero today, right?
I don't, unfortunately. I don't. I could mention one woman that I worked with for years at Columbia as a technician who probably has risen in my mind to the level of a hero. Her name is Kathleen Hussey. And if you just write that name down, Kathleen Hussey, the next time we meet, I will discuss her career because I think she was one of the first women to achieve a PhD in the field of parasitology. Got a degree at the University of Michigan. She was Dr. Harold Brown's, I don't know what her official title was. I think she was senior research scientist or something like that. She managed all of the teaching materials, training the technicians for this hospital, training research technicians, et cetera. She was a very hardworking, very knowledgeable, very humble person. And the more I thought about the work that she did after I graduated and went on in my own career, the more I realized how diligent and how attentive she actually was. And she deserves to at least be talked about because I think she kept her own personality underneath a large bushel basket. She was very shy. She never asked for anything. And Harold Brown was very, very gracious to her and realized her position because he was the only person standing between her and the university saying, well, we have to save a little bit of money and we have to cut back and that sort of thing. We've seen that happen time and time again. She lasted until the time that she retired at Columbia. And she was a gift for anybody who just got there and didn't know what to do. She would sit you down and show you the ropes and show you where your microscope was and where your slides belonged. And if you were training to be a diagnostic technician, for instance, she was a marvelous person, a very, very quiet person, though, I must say. So didn't garner a lot of attention. Harold Brown was a little less quiet, a little more gregarious, a little more outgoing, and very knowledgeable, of course, in what he was doing. But he couldn't have done any of that wonderful teaching unless he had stuff to teach with. And she was largely responsible for maintaining a very large teaching collection, in addition to her training duties for lots of other people. So in my mind, Kathleen Hussey, Dr. Kathleen Hussey, is a hero.
All right. Thank you, Dickson.
I'm glad you bring that up, Dickson, because it reminds me, and I always like to be reminded because I forget things, brain fog of the COVID pandemic. But we've been doing the fundraiser, right, to help the Peace Corps HIV and AIDS. But February 11th is a special day. I don't know if anyone knows. That's the International Day of Women and Girls in Science. This is a UN adopted resolution. We're going to continue, right, this fundraiser through the end of this month. But then we go into February, we're looking to try to come up with a way to support women and girls in science. So, people write in, give us ideas. We're looking for an organization to work with to help further that cause. Because I think as you bring up, we have a history that we need to correct of women and girls struggling to get recognized for all the great accomplishments and contributions that they've.
I would totally agree with that. There is a women's parasitology group, by the way, Daniel. I don't know if you're aware of this. I just found this out. There's a whole network of women that are working in parasitic diseases that have formed a large working group. And I will send you the email that I got from somebody, and we can share that.
I think it was from me. It was from me, Dickson. But
it was from you. That's where it came from. Okay, I was really surprised. Yeah. I had no. Let's make them our hero next time. Let's for the whole organization.
All right.
That's TWiP 191. You can find the show notes at microbe.tv/TWiP. Send your questions and comments to TWiP at microbe.tv. and your guesses, of course. And if you like what we do, consider supporting us. Microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center and parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dickson DePommier is at Triconella.org and thelivingriver.org. Thank you, Dickson.
Thank you, Vincent. Thank you, Daniel.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. Hey, Dickson, why don't we have a Parasitology Society support?
I don't know. I'm retired, remember?
It's not an excuse, Dickson.
No, it's not a good excuse. It's not a good excuse.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.
Parasitic.